So hi, my name is Tim Johanssen, and I'd like to thank Melanie Witt and Mark Dallas for the opportunity to come and introduce to you the Drug Discovery Institute at Oxford. It's the Alzheimer's Research UK Drug Discovery Institute, or as we like to call it, the ODDI. And this is housed within the Nuffield Department of Medical Research building. This is over at the Old Road campus. This very impressive new building over there on the site. And so what we're endeavouring to do there is to take academic research that's at the what we say is at the bench, and then translate that into a therapeutic that we can then hopefully progress through collaboration with pharmaceutical industries through to a therapy for Alzheimer's disease. So I actually began working for a pharmaceutical company down in London some time ago now, about 15 years ago. And at that time, there was only actually 4 drugs on the market. And so this company that I worked for, they make donepezil, or you might know as Aricept. This is one of the major treatment. And so it's of a class of drugs called acetylcholine esterase inhibitors. And at the time, they were all of that same class of drug. And what I want to emphasise is that since then, there's only actually been one other approved drug to come to the market, and that's memantine. And so what I've put up here is a diagram of the clinical, preclinical, and the outer ring, and the clinical stages. And this is a snapshot back in 2010 of the drugs and where they were at. But what we can see today is only one of those has actually been able to progress all the way through to phase 4, which is being approved for therapy. And so what this has led to is an exodus of pharmaceutical companies from this really important preclinical research because of the risk that's involved and obviously the financial cost of this. And fortunately for us though, Alzheimer's Research UK has identified this major problem in this preclinical phase of drug discovery. And in order to address this, they've established these 3 drug discovery institutes that Robin referred to as earlier, and they're housed, as Robin said, at University College London and at Cambridge, and here at the University of Oxford. So these, the Oxford Drug Discovery Institute wants to combine the academic excellence at the University of Oxford and the researchers here, as well as with the expertise coming from from the pharmaceutical industry in hope to discover new treatments for dementia. So they're set up somewhat like a pharmaceutical company is in the sense that they have a chief scientific officer, and then underneath there's a lead biologist, lead chemist, and then biology team and chemistry team. And then we collaborate directly with academics here in Oxford and further afield on specific projects that they've actually validated in their own labs, and they've shown to be efficacious in preclinical models at the very early stages. And then where the Oxford Drug Discovery Institute steps in, as well as the other institutes, is we want to take that basic at-the-bench research and develop high-throughput assays. So we want to take this, say it's a protein that they've found, and we want to come up with assays where we can study the effects of that assay, that protein in high-throughput sense. So that is so that we can study we can test tens of thousands to hundreds of thousands of compounds to find one compound or a class of compounds that actually are able to prevent the negative activities of these targets that have been discovered in the academic laboratories. So today I'll just describe 2 projects that I work on, both in collaborations with lead academic scientists at the University of Oxford. So the first project I work on is a collaboration with Professor Richard Wade Martin's lab, who's down at the Department of Physiology and Anatomy and Genetics. And in their previous research, they've actually taken one of the key mutations in Parkinson's disease, and they've been able to express this in a transgenic rat model. So they take a human protein that's abnormal, and they express that in a rat model, and then they study this rat model to see what the changes are. And one of the major changes they found was there's this normal process that occurs in the cell. It's a requirement for cell survival, and it maintains a balance between the manufacture and the breakdown of cellular components. And so we've just got a little picture here that shows the cellular components. And then you have these intracellular vesicles. They're called autophagosomes, but just vesicles. They come along and they engulf, this is all within a cell, they engulf these cellular components. and then they fuse with another intracellular vesicle called a lysosome, and this creates an acidic environment, leads to the degradation of these intracellular components, and then these can be recycled and used again. So this is ongoing at all times, and it's vital for cell survival. And so what we want to do, so with this discovery that when you have this mutation in these rats that are expressing this human protein, that this autophagy is prevented. This means that what we have what they call as a phenotype, so we can study this prevention of autophagy, and we can set up assays that screen, as I said, tens of thousands, hundreds of thousands of compounds. So, and then we look for compounds, preferably, generally in about 1% of these will show activity in preventing this phenotype. So they're called hits, and then they can provide us with a starting point that we can then, with work with the medicinal chemists in the institute, go about refining the properties of these drugs that have come out. So this project at the moment is in its early stages, and so it's just been developed from what the research in Richard Wade Martin's lab. We've been able to take that from a very low throughput system and miniaturize it, it's called, into high throughput scenarios. So here we actually take neurons, animals from these animals that I referred to, and then we can grow them in these little microplate dishes, and they have 384 wells on them, so we can screen on one plate, we can screen up to 300 odd compounds on a plate, and then using state-of-the-art technology that's housed down at the Department of Physiology and Anatomy and Genetics with its high-content imaging, and this machine here is called an Opera Phoenix, and it's definitely a state-of-the-art technology. It allows high-resolution as well as high-magnification images of these neurons that we're growing from the brains of the animals. So here, or nerve cells, and so here we can see them, and using antibodies we can stain and highlight the neuron in red, and its little extensions, the neuritic extensions that join up with other neurons. But more importantly, I don't know how the imaging is up for you out there, but you can see all these little dots. And so those dots are coloured yellow and green, and they're stained with yellow and green antibodies, and they detect those vesicles that I was referring to. And so with this image, we can actually use sophisticated software to really extract this information about this autophagy. So here you see a very complex image with different types of cells. In there in red you see the neurons, and then there's all these little spots which are the intracellular vesicles that we want to measure. So using this really extensive pipeline on the software, I'll just skip through it. We can find the nuclei, so that's the centre of a cell, the heart of a cell, and so we can isolate, but they're not necessarily all neurons, so then we can just detect the ones that are neurons, and then we can further isolate the region of the neuron and the extensions that we want to look for intracellular vesicles. And then we can detect all those little vesicles within there, and then what we're looking for is when the vesicles overlap, so that's when they fuse, and that's our readout of autophagy. And so we want to look for drugs in this screen. So this is still, as I said, in its early development. We haven't actually screened libraries yet, but we will hope to do that in the near future and look for compounds or drugs, whichever way you want to refer to them, that increase the number of those dots, basically, the overlapping vesicles, and then that's a sign that it's overcoming this prevention of autophagy. So in my second project that I work on at the Oxford Drug Discovery, we collaborate with Professor Sir Simon Loveston's group. They're in the Department of Psychiatry and also have laboratories down at the Department of Physiology, Anatomy and Genetics. And in their previous work, they've made a discovery that shows how important a certain protein named DICOP1 is in Alzheimer's disease. And along with other laboratories down at UCL and in Italy, there's been a wealth of research that's validated this protein as a target, a therapeutic target for Alzheimer's disease. And so we've collaborated with the Loveston's laboratory to further investigate this. So some of the further background is that the reason this is so important is, it might have been referred to before and you might know about it, but in Alzheimer's is the amyloid plaques. So this is made up of this A-beta peptide. And so what they've shown is that when you increase the levels of beta amyloid, you actually increase the amount of this protein DKK1. And this doesn't happen in the cases where there's not Alzheimer's present. And so this offers itself as a really appealing target for us. It's also been shown in animal models that if you block this protein, its activity, so when it binds, it has a toxic activity. So if you block that in animal models, you can rescue the health of these cells. And so that's what we wanted to do. We wanted to set up a similar system like that to screen for drugs that block this DKK1 protein. And so this project is a little bit more developed. We've actually been able to set up primary high throughput screening. And so I just wanted to demonstrate some of the equipment that we use because, for one, I'm quite impressed with it. So here's just a little cartoon of trying to just to demonstrate how you have this protein. In this case, it's called a donor, but it's our DKK1 protein. And when it's in close proximity to the binding protein where it conveys its toxicity, what we can do using tags is that will transfer energy that we can measure. So when they're close together, we get an increase in fluorescence, and we can measure that on a spectrophotometer pictured here in many plates, screening tens of thousands of compounds. So when we find a drug that blocks this protein-protein binding, the fluorescence will decrease. And so we'll be able to measure that, and then we'll be able to find out which drug is doing that. And the way we're able to actually handle the logistics of tens of thousands of compounds is made possible by using such technology as shown here. And this is a ECHO liquid transfer system, and it's a roboticized way of dispensing the compounds from a plate into the experimental plates. It doesn't actually sound that interesting, but it's more of the concept behind it. And what it does is that it actually uses sound waves to send nanoliter droplets, 2.5 nanoliter droplets, minuscule droplets, through the air to a plate that you want to do your experiment in that's upside down above it. I find it quite amazing. And so we use this to aliquot out our libraries across all these many, many plates as pictured here, so that we can then perform these experiments. So from these experiments, we've screened library of more than 10,000 compounds now. This is at Christmas, and we've found 52 drugs that are hits that block these two, the DKK1 protein from binding. So now we've taken these on, and at the DDI, we've developed secondary assays, and these assays are in cell lines now. So what we want to do with those is to validate these hits that come from the previous assay, because these experiments prior were just done basically in a test tube. Now we're putting them on cells that are actually growing, and we want to see that they're able to do the same thing. And in order to do that, we've developed assays again using this fluorescence spectrophotometer, and we can measure when the protein is no longer binding by the reduction in fluorescence. And we can also image it using this high content imaging that I introduced before. So again, we've got a couple of assays we can see, and we can show that the drug is actually stopping the binding of DKK1. And from there, from screening the hits that have come through to this stage of the project, we currently have 4 drugs that have been effective, but we have many more to test. So at this stage, this is where the project is up to, but just recently in January, we've been successfully in achieving one of the goals of the Drug Discovery Institute, and that is to form a collaboration with a pharmaceutical company in order for them to inject funding into the pipeline that I've just described, so into that first, the high throughput screening, and then the secondary validation screening in cells. And then with the aim of taking hit compounds through to animal models, so that by 2021 we can have a selective and potent drugs that can enter the brain, so get across into the central nervous system. This is a key problem with a lot of pharmaceutical products. And then that they also actually show that they can treat the symptoms of Alzheimer's in models of that, normally in mouse models. And then at that stage, the pharmaceutical company, so this would be the end of the preclinical stage, and the pharmaceutical company then would have the right to acquire these drugs, and then they would fund the incredibly expensive process of clinical testing, and with the goal of getting it to market. With that, I'll just finish and I'd like to just introduce the people that I work with. So John Davis is the CSO and the Chief Scientific Officer, and over at Cambridge it's John Skidmore and Paul Whiting. And Ellen Daniel and Paul Brennan are the heads of the chemistry and biology, and then there's a group of us working at the Institute over at Old Road, and we have our key collaborators as Simon and Chaz and Richard Wade-Martins, who I've just talked about, and our other collaborators, academic collaborators that we work closely with. So with that, I'd just like to say thank you.